Hi everyone, my name is Elijah. I am a second year PhD student at the University of Sydney, and today I'll be talking to you about my first year PhD project. So, by way of introduction, imaging by cytometry has really changed how we look at cells. We're now able to have higher resolution when looking at cells, which enables us to capture expressions of multiple cell types in intercell cell states. And doing so has enabled us to answer much more complicated questions. However, a gateway to answering this question is through clustering, by which we find similar cell types and aggregate them together. And for cytometry data, flow cytometry, there are a lot of methods that have been developed. However, for imaging mass cytometry, not so much. And most times, practitioners tend to use methods that were developed for suspension technologies like flow-sum and phenographs, which are common examples. And when clustering cells, I'm a bit clumsy. When clustering cells, defining distances between cells is quite important, and oftentimes the most common one used is Euclidean distance. And this may not always be appropriate, and we can see why. A few years ago, there was a paper that was released that looked at the effect of different distance metrics on clustering, and they focused particularly on single cell, and they showed that when clustering single cell data using correlation-based metrics actually outperforms the typical Euclidean-based one. So we had the idea that maybe this is the same thing for imaging mass cytometry data. And as with other technologies for data generation, there are problems that are inherent to it. So this is the image of a cell. As you can see here, there are some bright spots, and typically, you will normalize, because this is an artifact of the imaging technology. It's not actually bright when you do the experiment, so you can normalize it out. But after doing that, if you were to take like Euclidean distances, it will weight the bright spots as the same as the non-bright spots, and this can heavily influence your downstream analyses. Whereas with correlation, this wouldn't be a case, right? So the aim of this project was to access the efficacy of correlation-based matrices on this kind of data, and if we can combine these ideas into a package that's easily accessible to the greater audience. So to illustrate why this might be the case, we looked at an imaging data set that was generated from a human lung tuberculosis. And so here I've provided the annotated cell types. So these cell types were semi-automated, where they cluster it, over cluster, and then they merge clusters based on marker expression, and then they annotate it. So there is quite a significant amount of human intervention in this clustering. We then picked a random algorithm like hierarchical clustering to try and see if we can recreate these cell types. And we cluster using two different distances, so correlation, Pearson, and then Euclidean. And we used the adjusted random index, which is a set matching algorithm that tells you if your two sets are similar. So a value of 1 means they're perfectly matched. Value of 0 means that there's no concordance between them. And you will notice that the values for the ARI here is quite low. This is because this was semi-automated, and because there is human intervention, it makes it harder for a computer algorithm to recreate it. So this gives her a good objective measure. And that being said, we can see that for the ARI, the Euclidean one is half as good as the correlation-based one. So even in this very hard circumstance, we're able to get a much better score by using correlation compared to Euclidean. The next thing was to try and figure out if this is just an artifact of one data set and one technology. Can we extrapolate to multiple data sets over multiple technologies? So we collected about, I think, 10 or 12 other data sets from different technologies. And we did the same experiment where we clustered using distance-based metrics and correlation-based metrics, and we rank their ARI scores. And here, 6 is the best performing one, and 1 is the least performing one. And you can see for all of the data sets, or most of the data sets, that the bottom 3 are always in the top. So objectively, across multiple data sets from multiple platforms, there is a theme that correlation-based metrics are actually doing better than a typical Euclidean. So that was for one algorithm. What if we try other clustering methods? So like k-means, or what more specialized ones like flow sum and finograph, which are often used for these kind of data sets. We see again, this pattern is quite consistent. It's not so much so for the finograph, and but even then, if you look at it, it still underperforms compared to the other algorithms that use the correlation compared to the Euclidean. So this doesn't take away from the common theme that I am trying to tell you. So after looking at these distances, an idea came up that maybe if we combine them, we might be able to actually do a lot better. So sort of a multi-view integration of the data set. So a simple experiment was to generate a Cartesian product of 4 distances or similarities, including the Spearman, cosine, Pearson, as well as Euclidean. You can see that for the cases where we combine the data sets, they often perform the best. So there is some value added in combining these distances together. So we established that using correlation is better, using multiple methods combined is even better. How do we package this into something that's easily accessible? So I would like to introduce to you field sum, which is a clustering method for imaging cytometry data. And the first step of field sum is to sort of get our data and project them onto planes such that the dimension has been reduced. Because if you're computing correlation, these data sets typically range in n number of cells of about 300,000. So if you were to compute correlations of that in a computer, it takes a lot of time and a lot of memory. So the first thing we did was try and reduce as much as possible. And one of the most fastest methods actually using this technology called self-organizing maps. We use texture data sets, generates a bunch of prototypes that you can map your points to. So here you see that for this particular prototype, this black dot, there are 3 red dots. So it means 3 points from the original data sets that maps to it. We can then use these prototypes as proxies and then compute these distances or similarities on it, and then combine them using multi-view integration, and then cluster those prototypes and then map it back to the original data set. So you're essentially linearizing your clustering algorithm, which makes it really, really fast. So that's good and all, but how does it actually perform? So I looked at, again, those same data sets and compared it with flow sum, which is quite often used for this kind of technology. And so here I report the ARI, and you can see that if you look at the pairwise comparisons for almost all of the data sets, flow sum actually outperforms flow sum. And again, these scores are low because these data sets were all, had significant human intervention in the cell type. So we're not just matching another clustering algorithm. And this is significant, the difference. So it's fast and it does work. So this is an addendum to flow sum. As with most clustering methods, you need to specify your number of clusters beforehand, and this is often an art more than a science. So we try to make this accessible to people. So we implemented a few cluster estimation methods in the flow sum package. And here I have provided you the relative error of these methods. So this gives you the difference between true number of clusters and the predicted ones. And you can see there are 2 that are closest to the 0. So 0 is perfect. So the discriminant and jump method are actually the best performing ones. Again, you don't have to use it. It's purely a guideline for this. And if you do decide to use it, it performs decently well at capturing the cell types again, right? So if they should just plug in your data set, say I don't know how many clusters there are, but I want to estimate it, and using those, how well do I recapitulate the actual scores? And you get something like that. And so this is, I am a computer scientist by training, and I like to make things fast. So I was curious to see how this algorithm scales in the number of cells. Because it's practical, I don't want you to be waiting like an hour for something to run. So I took a data set that had about a million cells and tried to see how long it would take me to run it to completion as the number of cells increases. You can see that for the number of cells, as you increase the number of cells by tenfold, the algorithm is actually almost linear in the running time. So for about 1 million cells, I was able to run it in under 5 minutes on my computer, which is by no means like a server that has lots of core and memory. And also, in terms of the memory usage is scaled linearly with the size of the data in gigabytes. So for about 1 million cells, it took me about 8 to 10 gigabytes of memory in the maximum. So to conclude, we've shown that correlation-based methods does actually outperform Euclidean-based ones when clustering this kind of data. And I have packaged this into a tool that's easily accessible and is available on GitHub. It's fast and it's robust. And that performs the current gold standard, I would say. So please feel free to try it and let me know if you have any thoughts about it. So, this kind of work is never done by yourself. So I'd like to thank everyone who was involved in me getting here. So my supervisor, who's sitting just over there, I'd like to thank him for his continuing support, and the Precision Bioinformatics at the University of Sydney, who's always, foster a culture of research that's amazing, and my funding source. And and you guys for inviting me here. It's been a pleasure presenting my work, and I look forward to questions. So if you are absolutely fascinated by self-organizing maps, I would love to talk to you about it. My poster is, I think, 166. Please come and find me, and we can talk as much as you want. But thank you again.